Hello, everyone. Welcome again to our Data-Centric Biological Design and Engineering Seminar, part of the Alan Turing Institute interest groups. So this is our final seminar of the year 2024 and 2025. And new talks are coming up soon, so please follow our website. I just pasted it in the chat box. And today we are very happy to have Professor Dr. Fabian Theis. Professor Dr. Fabian Theis is a professor at TUM School of Computation, Information, and Technology, and now leads the Computational Health Center at Helmholtz Munich. Fabian's research used artificial intelligence to uncover the secrets in human cells and has developed a lot of useful methods and techniques, particularly in single-cell analysis. And today's talk will be on modeling single-cell states and dynamics. Let's welcome Professor Dr. Fabian Theis.
Yeah, thank you so much, Yushin, Pedro, everyone, for the nice invitation and the possibility of being able to talk to you today. I'm going to share my screen and
Yeah.
Maybe you could just allow me to.
Can you share?
Now I can. Appreciate it. Thank you so much. So I guess you should see my cover. So I want to talk a bit about how we use Gen AI to essentially learn then state and dynamics as well as response. So that's sort of what I summarized by that. I said before, I have something like 45 minutes, but it can be shorter or faster. But particularly, we would really appreciate if people have questions, just like unmute and ask in between if you want. I always find this like talking for 40 minutes type of thing. Yeah, maybe you might drift out and might just like do something else. So if you want, yeah. And I'm going to talk about design a bit, but just try to motivate where that design comes from. We had some really exciting results, I think, on basically an organoid protocol design type of thing. It's like something where we really carry this through at scale where, yeah, I'm just going to show you in a minute. So I want to start with the Gen AI thing, right? Feynman, famous physicist, you know, what he cannot create, he doesn't understand. I think it's really nice saying from physicists that we want to have these forward models, right? These forward models that allow us to, yeah, then sort of maybe predict also, like really extrapolate. And I think with Gen AI, we might be sort of be strong at being within interpolation, but maybe you can also go beyond this. And I think this is maybe what you guys want to do with design as well. I just have one slide on our place. Just because I think there's a whole bunch of opportunities for exchange. We have a series of exchange programs, and there's so many of my people going to UK in particular. There's like this I'm from a technical university as well as Helmholtz Munich. We have some specific programs with Imperial and so on. And essentially work sort of on all kind of omics, big data, and this computational health center that we set up here in Munich has been now sort of as mission as maybe this one Turing branch has sort of develop apply AI for discovery and translation. And it's been really growing. I think with now something like 500 scientists and more than 40 PIs. We, I think, probably the largest place for this type of sort of health AI type of research in Europe. So I think it could be fun to think about angles. Yeah, this is just some numbers what we do. We have a bunch of grad schools. This is this Alice network that I was mentioned before, Yushin Pedro. I think it's a really nice sort of conglomerate of AI researchers, whereas also exchange programs. We have a lot of clinics and pharma partners and AI partners. Right, so that's just my little thing about the place, because I think it's always interesting to see what different places are. And I'm gonna definitely check out your, how do you call it, Yushin, interest groups, right? I'm gonna check that out. So I want to talk about single cell analysis, right? And we have this old sketch that we did in a perspective some time ago, that these cells. We've been always trying to understand cell fate, right? But I think sort of this picture of maybe what development biologists would call Waddington's landscape, like a cell going through healthy aging and maybe sort of then taking a wrong turn. That's something we want to model. And I think the aim of pretty much my talk, but a big part of the field would be like we want to learn this landscape, like quantitatively, but then not just be able to learn, but act upon it, right? So the learning part would be, we can reconstruct trajectories because we have so many. Typically not longitudinal, but we have so many samples. That could be transcriptome, could be other type of information. But we can build these things now. Trajectory learning, this has been something my lab and many others have been doing for some time. But we might be able to see also where these things sort of go awry to the disease. We might be able to tell, because we sort of have very molecular, sort of quite causal type of observations potentially, what type of perturbation we need to do to go back to the healthy case. So that's this learning and acting upon. And I think I guess you might call it then design, like, what's the perturbation to choose, right? So that's sort of a long-standing dream. This is obviously just an artist's sketch, but we have actually real of these landscapes. So in this case here, we are flying, and I hope the movie is working, through a landscape that's been reduced from 20,000 dimension transcripts to 3, where each of the colors sort of indicates different cell types in the murine gut. This collaboration with the Lickert lab, where we see these intestinal stem cells over there that sort of turn into other stuff such as enteroendocrine cells, like different cell types in the gut that you need. And you saw they weren't just sitting randomly in this space, they were sort of occupying specific places, the same cell types obviously being close by. But here, for example, there's this bridge, a progenitor cell type. And we sort of had some very exciting questions that we were able to answer in this thing. I won't sort of go into details, but this learning of trajectories, this was some of this really early work of, diffusion maps, sort of more complex processes, and essentially sort of just trying to understand these type of connections between clusters, if you want. We've been actually extending this, so if some of you are not interested in cellular relation, we've been extending many of these ideas also to other, yet just transversely observed longitudinal processes. In this case, for example, each of the dot would be an eye of a healthy, or of a patient, a healthy subject, with a disease here in the patient case called diabetic retinopathy. And you see, if you sort of arrange them, you sort of see this transition through different disease stages that we were able to model as well. And we've been actually sort of putting this model into a toolbox. Just recently got that even on Nature Medicine cover, even though it was sort of a computer type of thing that would allow you to sort of track then these journeys, not just in the cellular case, but also in the patient case. So if there's some interest, happy to discuss it afterwards, but I just focus now on the patient thing, on the cell thing. And I want to have this one more point, is that so much of, I think nowadays, AI anyway, but also of this sort of particular computation biology effort is engineering, right? Early on, we set up this toolbox called ScanPy, that became one of the two sort of de facto standards for single cell analysis in the field. But at some point was too big. I think it has something like 5 million downloads now, too big for us to handle. So we set up this consortium with a lot of international colleagues and friends that now is this whole package of ecosystem, Python-based single cell analysis, particularly attaching very well to the machine learning ecosystem, obviously. And we've been doing conferences for that. We've been extending this to spatial omics, a big trend, Nature Method of the Year twice now in the past 4 years, where you can sort of have spatially resolved single cell multimodal observations. You do some analysis in that. We did a quite popular toolbox on top of that. We came up with a fair data framework for doing this together with the Stegler lab and the Moore lab. And these type of tools, this sort of engineering stuff that lies at the back, is just so important, so that your lab can share these things, but also your partners sort of have a standard thing to then do stuff upon it. We run NeurIPS competition on top of this stuff, but this thing needs to be sort of fixed. And most recently, we've been going also in perturbation analysis, representation of many, let's say a drug screen or something like that with a single cell readout, and sort of putting this together. So some technical work, but I think exciting and useful to have downstream. So happy to connect if you're interested in this. And please, if you're someone interested in single cell, join this SCVerse. It's an open and really friendly community. Well, yeah, and we talk about best practice. So if you don't know so much about it, we've been writing this online book, Single Cell Best Practice. It has been sort of really cited and used a lot, but we have also sort of this review about it. All right, so let me get started. What's the AI part here? Because you guys may be more interested in this part, right? So I think the very simple thing is we have the gene time cell matches. We don't really care about a single cell. We don't have much more information than this sort of multidimensional transcript information that you get from, let's say, single cell transcriptomics or multimodal observation. My lab, in parallel with Neil Yosef's lab, introduced this concept of unsupervised deep learning, I would say, where you sort of now take them and you just run them for, this time, often just an autoencoder. So you squeeze them down, blow them up again, make the output as similar as possible to the input. If you do that, because you sort of encode them here, this low dimensional space, you clean up stuff. But what's interesting is then, this bottleneck, there you see some of the dynamics, some process, some actually interesting information. And this was something that we've been really using ever since. We've been putting constraints upon it. We add sort of various biological priors, let's say programs, dynamics, things like that, and it really gets interesting. I show a few more examples in this direction, but the key one I want to sort of interest you here now is what happens if we have multiple of these data sets. This is sort of a key thing. We want to build a human cell atlas, let's say. It's like multiple labs contributing to the data set, but often then you have this massive batch effects, as you always have in these big data sets. So then you add this information here, you tell a method about the data set, and then it can use it to sort of superimpose and more efficiently encode them in the bottleneck, just a standard conditional autoencoder, conditional rational autoencoder, and then you clean up the data. And we've been running a lot of competitions. These things are really working well. We've been having some sort of a bit more causal, at least sort of disentanglement type of extensions on it. This is exciting stuff from Army, I think, where we've been now taken the same variational autoencoder, actually conditional variational autoencoder you saw before, but we've been now sort of cutting each of these individual dimensions in subgroups and then individually decode them. This is called additive decoders. There was a recent Europe's paper upon it showing disentanglement. We've been extending this to sort of a nonlinear aggregation pooling here, and we can actually theoretically show if you do this process, and this is of course a much simpler type of decoder than you had before, but in this case we can actually guarantee disentanglement. And we see that the expressibility of this thing is quite strong. So with this disentanglement, suddenly, you can go to, in this case, now this would be different dimensions. Suddenly you sort of pick up in each dimension a cell type or something like that. So this is really a useful thing for sort of more interpretable latent spaces, which typically they are not, right? All right, so let me show you what we do with these things. So this is work by Lisa and Malte, PhD and postdoc now moved on to PI actually in Helmholtz, where we built this human cell atlas, a lung atlas. Took a bunch of data sets and annotate them, harmonize the information, and essentially sort of just build this big map. So just taking all these data sets, doing the embedding as I showed you before, and then identifying different cell types by markers and jointly annotating with a really large scale consortium, many partners here contributing. And to make this sort of look more like an atlas, we put a circle behind it, and sort of looks a bit like the continents. Bit of a standard joke that I do, but then if you zoom in, you sort of get all these additional information that you can now use to annotate your next data set, to sort of find information that's sort of coming from this big community. And this is what we deliver in the Human Cell Atlas. So you have this big background thing. But what you can now do is now you can map disease on top of this. So one thing we, for example, found, there was one cluster that's high in a factor called SVP1, which is sort of associated with some type of, I think, cytoskeletal things in cells. And this is in monocyte-derived macrophages. These are actually quite interesting then shared across COVID-19, lung carcinoma, and I think IPF or something like that. So this is kind of really excitingly shared thing that you initially wouldn't connect. But now you see sort of a shared thing, and you could sort of then maybe look for shared targets, drug repurposing, this type of stuff. So that's the use and attractiveness of these atlases. And let me show you one more. Oh yeah, we have a best practice on this, but let me show you one more atlas. And I want to sort of use this as an example for perturbation modeling in a minute. Together with the Barbara Treutlein and Greg Hamplab from ETH and Elizabeth Binder's lab, we've been building a human organoid atlas, particularly for neurons, for brain. So these organoids, they sort of reflect actual development. We've been but do this in vitro. So we've been aggregating a lot of these across different time points. We're putting them again in this big map that you saw before, but now everything is sort of more smushed together because, they're just sort of growing, right? And you see here age and other things sort of put on top. And there's different ways how to make them. And you sort of can look for your favorite marker. If you were a biologist, you would sort of see, hey, where's my thing in, let's say, non-telencephalic neurons or something like that. But what's exciting now, and I think this is sort of a cool transfer learning type of challenge. Now we have this primary atlas. This is brain development, fetal brain development, okay? And you can ask, hey, how does this model system differ from the primary atlas? And that's now a design question, if you want, experimental design question, right? Like, where is this thing not yet enough? And this is what we did. So we just mapped this organoid one to the primary. And then we, for example, just compare what's different. And you see sort of there's a bunch of pathways downregulated in the organoids. Like cell adhesion is not as good, but there's upregulated some type of sort of ATP synthesis because they need more energy because they don't have any innervation or something like that. They don't get any vascularization. And what you see is across these different organoid data sets, this glycolysis score actually differs. Some of them are sort of better than others. This tells you that the way how to make them actually sort of influences some of these covariates. And this is what we actually more systematically now did. So we take the primary atlas and we map on the primary one by color the organoids. And then with blue, we say, hey, yeah, this is what we capture in the organoids. And with red, we say that's what we don't capture. And if you now zoom in, you, for example, see here a bunch of cell states that you actually just don't capture at all. These are non-neuronal cell types, and this would be just, I think, immune cells or so that just the organoids don't do. That makes sense. But then there's a bunch of others, and then you sort of look across all the clusters, and then let you see that there's, for example, some sort of midbrain, hindbrain, and other states that are being generated here, right? All right. So just to summarize that first part, we built these organ reference atlases by using these, I mean, they call it AI-based, but this variational autoencoder early on, later transformer-based integration, and then you use them for mapping. If there's any questions, happy to go into that. Otherwise, I just go to my next part. Okay, fine. Did you see my fantastic animation? So now we have data set, right? But like what's the model behind it? There's this idea, hey, can we build, this is an illustration from CZI, can we now build a virtual cell from that? Can we integrate a lot of these data sets to potentially then build these integrated. Okay, so how do we act on these cell landscapes? Like there's this traditional thing of what's called whole cell models, where in systems biology we try to do this with physical and chemical representations, and you sort of predict the phenotype, but like you try to sort of put all of these compartments together. It kind of worked, but nah, this thing is not really predictive. People talked about virtual cell biology, but quite frankly, I think all of these models, they weren't really actionable, right? So that's sort of a problem that we then sort of, people sort of got disattached to this. But now, recently, colleagues such as Demis or uh, um, people from CCI, particularly Jan Max Zuckerberg, so talk about this virtual cell as another thing again. So this presentation, we actually put all of these information in one big map. It's really getting attractive. This is from a review we did recently with Raoul Satici's lab. If you take maybe even multiple modalities, you sort of chuck them into one reference. The virtual cell could be this type of reference here, because these cells now sort of integrate. They don't really fully exist because they've been sort of merged, right? But now you take a new data set, you map them on top, and you get sort of this information, just as we did before. But you could sort of push this even further, just going beyond references. This is a big review we did, like a community thing led by Stephen and Emma, where you can't just don't do only reference, but you might sort of even learn this latent space dynamics. You might learn effect of perturbations and so on. So it could be built this one foundation model that factors to all of these downstream tasks. Typical type of question, right? So this virtual cell is sort of this digital twin concept. It's like an old thing, right? So we've been thinking like, how can we do this? And traditional ones is just really based on first principles, very old school type of approach. Put all the laws in. I think this kind of failed in our discipline. That's what I showed before, right? So now, there's like the other alternative. Let's go fully data-based, right? And there's another example. You test that as many others. Sort of just integrating all the data set. Let me show one example where we do this before we sort of talk about the third thing where you sort of do a hybrid version of that. So I think one of the most exciting things to use these models is for predicting perturbations. And early on, there weren't much data sets, but now this really grows. This is from, for example, a company, there's many competitors now that sort of really do at scale an experiment. They did like a single experiment and did more than 10 million transcriptomes. This is like huge, right? So they did more than 100 or like 100 treatments or something like that across many samples. So it gives you like this really rich space of effects of perturbations. There's like a competitor company doing this, CCI, Vivo is actually releasing this whole data set, and there's many others. And I just want to sort of say our toolbox, the ones that I mentioned before, is now ready to work also on that scale. So they work on more than 1 billion cells, essentially just using out of core analysis, really pushing the tools towards that. But now that you have these big data sets, like how can you fact of a perturbation? This is my little visualization, but I always like this because this was which is that broke the internet with respect to Gen AI. Remember the Pope wearing this white down coat, which is, can we, like, how is, and then just subtract how these things work, right? So that's how we go to new states. It's kind of like a similar metaphor, I guess, what we do with these cell perturbations. Let's say we have a cell actually observed under perturbation. It could be a drug, but it could be CRISPR knock measure, the idea of in silico perturbation as well. I'm just going to talk about the RNA-seq ones in the next slide. But if you're interested in sort of other modalities, these things usually generalize quite a bit. So it's this in this simple plus minus thing is essentially like a latent space arithmetic type of approach, but since then, many more fancy approaches came out. Most fancy ones is, in computer vision, if you really want to generate realistic effects of perturbations of images, sort of take inference, you just guide it towards what you want. So this is the idea that we try to use now in our cell setting. Now, cell setting for images, but essentially for these vectorized, more or less tabular type of expression profiles. essentially, you have like here an uniform Gaussian noise or like a Gaussian noise type of thing, and you sort of get to this bimodal distribution that sort of makes something interesting. Of course, the way how this is done mostly nowadays is not this stochastic way, but a flow matching, sort of this more deterministic type of approach, and this is what we're actually going to employ here. And what's sort of a bit of a challenge in our field is that we don't have these matched images of, without, but we just have like a whole bunch of different people, and we need to add classes without sort of knowing what the matched people is. Because like typically whenever you have a cell under perturbation or not, you just have it once, the cell is dead afterwards. So you need to do a distribution type of matching, and we use some version of optimal transport for that. But in general, it's now like, how can we do flow matching for cells? So this is our idea. We can actually, one of the examples is going to be fate engineering. Remember, I talked about those brain organoid protocols. Can we now essentially come up with an optimal protocol to generate some of these sort of mid or hindbrain type of cell sets that we don't find? This is sort of our motivation. But of course, this goes for many other type of things. You can look in development, you can look in knockout or drug treatment predictions. So that's like the whole battery that you can do. There's like 3 challenges that I want to sort of briefly say how we got around it. So first one, of course, is this unpairedness as I was mentioning before. So for this, you need to use OT to pair cells. And this has been tried quite a few times now in the field. We recently came up with this toolbox Moscot that is actually a multimodal single cell optimal transport type of approach with both spatial as well as temporal type of mapping things. I think quite cool and useful. Check it out if you haven't seen. Very scalable because we collaborate with Marco Couturi's lab, who really knows how to sort of scale up some of these algorithms. And this is like a baseline thing that we now use to pair cells. We want to have combinations of perturbations. There could be very different ways what combinations would be. For that, we use essentially a tangent, like a transformer-based embedding, to aggregate these perturbations, like a general perturbation representation. The last thing is we want to have these realistic cell profiles, not just these sort of rough approximations as these autoencoders typically did. Of course, for that we use flow matching, as I was saying before. So this is what we call cell flow. This is work by Dominic, PhD student in the lab, and Jonas, who is in Gray's lab as well at Roche. Yeah, and I just want to show you a bit what this can do, okay? So the idea of the general perturbation embedding could be, perturbation could be a drug, could be a gene knockout. Later, we also want to do this for genetics. There could be different dosages or timings, and there could be covariates, and we put all of them together with attention. You could just like stack them, but I think multi-head often works quite well. Well, and then you have your control cells perturbed, you match them with OT, and then you sort of guide the flow by this condition, right? That's the idea how you do it at training, and at inference, you sort of extrapolate. So just sort of to do this again, a bit more formally, you'd say you have your position in space, condition, and the time. In the encoder part, you sort of go in latent space, decode, you sort of decode to get your flow. And during training, then, you sort of couple them via OT, and then you let them flow. And at inference, you sort of just have various time points, you push them forward. And there's various sort of more fancy extensions that you can do to also sort of include this to stochastic, but this was sort of what actually already worked quite beautifully for us. So let me show you a few stats for that. And there's a bunch of data sets that I don't now extrapolate on, like really sort of go into detail what they are. But this is, for example, a drug perturbation data set where we can actually go out of distributions. We always hold out a few of these data sets. We go out of distribution. Then we see that, or we go out of distribution with a dosage often. And then we see some baselines here, but higher is better in this terms of R squared. Cell flow is doing very well. We can also make like an energy distance, distributional type of distance. We can do this for combination of drugs. We recently did a combination data set. And here it's interesting that we outperform sort of very simple linear baseline models, additive ones, which often models weren't able to do. Particularly these sort of foundation model-based ones are not yet on that scale. And then we also do genetic perturbations where we see that in this case, we also do well job. And last but not least, actually also we go on image-based data. In this case, it's for eye, and you can do it for other ones. So this is sort of good baseline that gave us hope, hey, this thing is doing something. We actually we've been sort of looking also at how this thing scales, and we actually see a bit of a beginning of a scaling law, which you kind of want to go, do we need more data, and this data is expensive. So we see this here for this data set I was showing before. You see the more cytokines we include for test donor, we see that there's actually a slight increase over time with cell flow. This works for donors. This actually also works. This works for number of cytokines, but this also works along the donor axis. And there's been a very recent exciting one called Tahoe 100. This is from this company Vivo, which actually renamed to Tahoe, which go more than 60K conditions. And there we also see sort of the more conditions we train, the sort of R rate goes up. And you're just sort of doing this training across 100 million cells. That was like a real challenge, and kind of happy that this worked out. So if you want to look at some of these benchmarks, there's the Cellflow papers out, and I'm happy sort of to support you with implementing and checking out. But let me now show you how this works for this fate programming, okay? And I don't show you yet for organoids, but I show you first for the neuron fate programming. So this is stuff from Barbara's lab, who has been sort of looking for a long time in taking iPSCs and sort of pushing up this factor NGN2, and it sort of makes neurons, right? And there's different type of programs that influence how these neurons being made, and then these things are called morphogens. And depending on which concentration, which combination you add, these protocols do different things. It's like a bit of an alchemy thing for me, but this is also maybe my naive biology understanding. But everyone has sort of their own type of protocol, and yeah, which ones do now what you want. So the cool thing is, this is a map of various combinations. Barbara's lab has been doing nearly 200 conditions, and you see sort of they fill up different things. We have a map so we can annotate them, and we see that they sort of also cover a whole bunch of brain regions. And here, sort of depending on which factor you get, you sort of see different enrichments of cell proportions. So that's a baseline to train on. And of course, the question is, can you search for all the other combinations that you haven't seen yet, right? So can you sort of play around? Can you, for example, hold out a few random ones? How does the model work? And what you see now when we do a hold out, we see that our model versus some of our earlier ones and competitor models sort of does a good job, as you would expect. It's sort of been pushing this. And we can also see that it captures quite interesting the covariance structure much better than other ones. In this case, for example, for combination of two factors. So now, can we now predict new clusters? For example, go in a combination that hasn't been seen. So we just take two factors that you could add here, those two, you hold them out. Like, do we generate like a really new state? And then we see actually that, this is sort of what the new one is, which doesn't exist in the old individual ones. So you see the individual ones don't give rise to the combination, but the prediction actually does. And this is also what we see. So we train on a data set. The ground truth would sort of be these ones we train on, these ones you want to predict. And hey, we predict this one. So that's exciting. So this shows that, this model doesn't just sort of interpolate, but it kind of goes to really new type of states that maybe more classical models had problems with. So let's go, and we can sort of look now at the score. We can look at the whole screen and we can see what type of predictions there are. So let's go a bit more crazy. Let's look at the organoid screen. So this was not just cells as it was before, but now we take a bunch of sort of different, there have been also some of these screens already where people try different modulators across different concentrations and timing. I mean, now we try to encode this protocol space, okay, this way how to make organoids. Now, not just now cells, but organoids. There's different factors that you can give at different time points. So this would be sort of timing, and you can sort of give them at different time points and different concentrations. This is sort of our space that we want to search for. This is like your biodesign space, if you want. And depending on this one, we can now just we hold up one data set, train on the other ones, and we see how well we predict versus baselines. In this case, we couldn't take any other models because only our model can do that. And we see we always outperform some simpler baselines. So now we train across all of them and do what we then want to call a virtual organoid protocol screen, okay? Our virtual organoid protocol screen. So for this, we take these different base protocols, add various combinations of modulators, and then in silico generate more than 23,000 protocols because we give them at different timings. Of course, we sort of still discretize that space. It's a bit of a random choice. You could, of course, make this more detailed, and you could potentially like make this fully continuous and then, go full RL or whatever on that. But for now, we just sort of exhaustively searched. Across these 23,000 protocols, we generated more than 70 million virtual cells, if you want. And now we can just analyze them as if we would analyze real ones. And we see sort of different proportions across these different factors. So it's kind of really interesting now to see, hey, what would those protocols do? So which one do you now pick? Well, for that, we thought, let's look what these different protocols give rise in terms of cell populations. And of course, these protocols come with errors. We could actually include them a bit more. But for now, we should have just quantify two things. First, we quantify how close is my protocol. Now, each of these dots will be a protocol and an ensemble of cells behind it. How close or how distant is it to the training data? So low E distance, very close, here very far. So these would be sort of really interesting ones. This would be sort of pretty much what you have in the training data. But along the x-axis, have how distant or close it is to the reference human data, okay? So this part here is interestingly close to the human real situation, but sufficiently distant from the one that it has been trained on. Now we can just like pick out a few, and then you see that sort of depending, like in this case, for example, if you give a lot of these particular factors at the end, you generate like more of these sort of neuroblast cells, and if you sort of change this up a bit, you generate different ones. And we are currently now following up with this validation. But this is a way how to do, I think, exciting experimental design. So to summarize that part, I think we can do rather stable but state-of-the-art perturbation prediction with Cellflow. We're quite flexible, but of course, limitation is, you need to have some large-scale perturbation data to train on. And we actually also don't integrate yet across studies. So that's my summary. I think with Cellflow, we can extrapolate cellular response profile. That would be summary for my second part. Yushin, I just continue?
Yeah, I think there are two questions in the chat box. The first one is from Wanjing, saying, a quick question, have you observed any cases where different or mixed modalities suggest different developmental trajectories? And if so, how do you decide which one to trust?
This
Yeah,
was
good
for the
one.
first part, yeah.
Oh,
Yeah.
thanks. Yeah, I should have made raised before. It's a good one. Yeah, I always talk about, like, also with this multi-omics single cell optimal transport toolbox Morse code. We've been actually doing multi-omics integration, and I think de facto at the moment most people just integrate on the RNA-seq because that's like the easy one to monitor than, let's say, ATAC or image. But rightfully so, of course, before you see an effect on sort of gene expression, chromatin needs to open. So typically what people see is that, if there's like a branching event happening, you see it a bit earlier in the opening of the chromatin, which you measure by ATAC-seq than with RNA-seq and similar else. So I think there have been a few papers showing that, of course, more information gets better. There's other information where you can add that's even like we're starting to do this with a few colleagues, like even single cell proteomics, but mostly marker-based, antibody-based protein information. And then you can discern immune cells more robustly. So even just for the clustering, it's a good idea to add more information, but then also for the branching, how to decide which is a truth, it just gives you an orthogonal picture often. There's like, you can get metabolism information. Then sort of this big cluster of your homogeneous cell type maybe then splits up by, being early, late in cell cycle or whatever, right? So yeah, I think it's basically, to summarize, it's always complicated. Yeah. But de facto, of course, most projection will be on the RNA-seq because that's something that you have mostly. Okay.
I mean, there's one more question. Can you and how take into account the dynamics of cells? This is a question by Jiyang Zhang.
Yeah, it's a good one. So in the cell flow, we actually have this as a covariate. So you can just like really briefly jump there. And it's a good point. So you see here we have perturbations, but we also have dosage as well as timing. So that you can sort of add it in and you can encode it actually as a bit more continuous. But yes, this might not be the most adequate thing, but it's a fantastic segue to the last part of my talk, if I may. So thanks for that. So basically what I talked so far was like purely database, right? And then of course there you kind of lose, it's like, hey, I need to read, I train on, let's say, observing when I throw an apple and I kind of need to find Newton's laws again, right? But maybe in hybrid models, can sort of put some of these first principles still in. And even though like I think in these very big modern AI models, we see that, you remember in the first AlphaFold, equivariance was a thing, but then you can sort of just train it from data, and could maybe these priors can at some point just find from sufficient large training data. I think at the moment we are not there yet. So I think hybrids is still going to be a thing. Actually, also Demis had recent discussion on that. And so I think it's interesting. And this is sort of what we do, right? And there's been a few reviews, maybe you can combine them. You can make everything differentiable so you can actually learn across like a prior and non-prior based one. What's the biggest prior we can put in? And you mentioned dynamics. For me, this is in particular transcription dynamics, right? Central dogma, RNA is being made, makes protein, is being spliced. And there's been a few really nice models for that. And very early one was called RNA velocity that was proposed by Stan Linus and Peter Kachenko's labs. And we've been following this up with a thing called SCVelo early on, essentially sort of modeling the splicing kinetics. If the more unspliced and spliced feeds you have, the more this gene is being made, otherwise being turned down. Put this together, essentially gives you like a vector direction for each transcript, whether it's being made or not. Put them together and it gives you arrows on this differentiation landscape. That's the idea, okay? So the question is, can you add that information to, let's say, perturbation prediction? Very good one. And this is kind of what we try to do here. I've been following up these early models with a rational inference, autoencoder type of thing. But these things are very good at the dynamics, but they don't use a regulatory model. There's been a bunch of papers, and I don't sort of give you the details now, that sort of add regulatory information to the process. So you put the GRN in, right? Because that's sort of what should encode the dynamics. But then they typically are good at telling you the next state, but they don't really tell you what the actual, temporal resolve dynamics is, because, finding parameters of these is like really hard. So what we try to do, we try to smush these two things together. So we have this very flexible embedding thing here, but we add the dynamics. So the dynamics would be sort of encoded in that forward thing, and we just write up this old dynamical equation, like how would a gene expression change over time according to a GNN pushed forward. But you have this huge parameter set. So it's like a 10,000 dimensional dynamic system. And in RNA velocity, we sort of solved this one by sort of decoupling it, completely ignoring regulation. But now we want to put it back in. So we call it reg velo, regulatory velocity, okay? And I think we actually have something working. This is work by Wei Shu, Ken Wang from the lab, a really talented PhD student that sort of takes these embeddings of spliced, unspliced reads, but also takes a prior graph. And we sort of embed these, and you'll get our latent time representation, but we take the prior GRN dynamics that we sort of estimate, but also are able to fill up, and then get a transcription predictor for each transcription factor. And then we put this transcription dynamics into the real dynamical system where we model transcription, splicing, degradation in this ODE system, but which is now coupled. So it's not a single one, it's not like single one-dimensional ones across all transcripts. It's a very one big one, which you need to sort of estimate parameters. But the beauty is, and this sort of, I think, Ken's main contribution is actually able to do the inference now in this thing. So we can do really joint parameter inference, variational setting, get a bit of uncertainty even there. And we have this 10,000 dimensional ODE that we now need to solve. So this works, and let me show you a few examples for that, of this approach. And now sort of the dynamics is fully embedded in there. So here we look at a data set. Early on, there's just cell cycle. And we have a marker of Fuji, it's like a marker that tells you about cell cycle states. We can see how much we reconstruct. And if we just take the embedding from Rigredo for this sort of cell cycle, for the cycling cells, you see, yeah, hey, nicely, we get the right dynamics as you would expect. Remember, we have arrows, we have vector fields you can project it on top. And we can also quantify this. So we came up with a bunch of metrics to quantify them, and we can compare these metrics of our approach via some existing methods, and we sort of do a very good job. We can quantify consistency as well as sort of this latent time, this data crosser, and we sort of really, by putting together these two aspects of regulatory network as well as dynamics, do a good job. But we can also look at the regulatory network that's being updated from the dynamic estimation. And we can see what sort of states we estimate here. And then we see also here, the model is doing a better job than existing approaches. So it sort of gets, that's what we hope, and I think we can show to some extent, gets you the best of both worlds here. Yeah. So we can now do self-weight analysis. Essentially, we take now the original network, but we can do in silico knockout because now we have a GRN on it, right? So with the GRN, we can just like change. We say, hey, this transcription factor is off. And we can then sort of just put that activity to 0 and can see what sort of the change in the dynamics would be. We can then look at the local effects, and we can sort of compare the dynamics, and we can sort of see what the cell fates, where they would eventually go, like this one then goes down or something like that. This is what we would see. And this is now what we actually can do in practice. Maybe I skipped this one example here. I just show you in collaboration here with Tatjana Sauker-Sprengler from Oxford, together with Xiuan Hu, at the time in Oxford, now I think in Wuhan University, where they've been doing both SMART-seq3 as well as 10x multiome across different time points in zebrafish neural crest development. Okay, so time development here. And she didn't only observe expression, but also expression under knockout of a few transcription factors. So we can really test the model under that. We just train an expression not on the knockouts. So we see first we get a latent time, and we can show that this latent time actually associates correctly with the different staging of this neural crest growth, which is nice. We had multiple time points. Those multiple time points fit to the latent time that the model estimates. But we can also see what the terminal states are that this model predicts. So where would those cells flow into? What would be sort of those wells in the Waddington's landscape? And they actually then consist to real ones that people would expect in this neural crest formation. We can also show that this model does it much more robustly than existing ones. And now, interestingly, we can now knock out stuff. Okay, this was sort of this concept of knockout that I showed you before. So let's take a bunch of genes that are known to be relevant for mesenchymal cell formation, and we see if we knock them out, always mesenchymal state, this head mesenchyme goes down. Similar for pigment ones, then the pigment ones go down. Now we look in the in silico ones, in the action, not no, in the in vivo perturb-seq, not just like stuff from papers. And then we can also see how well our model estimates the effect of the perturbation, and it really blows out existing approaches that don't use this dynamic information. So we're really excited about that. And then, we were able to formulate some particular hypotheses that I won't go into detail. So to summarize that part, I think with the dynamics, with the hybrid modeling, we can really add this GRN information, and it kind of gives us information about the perturbation dynamics. Of course, it's going to be very hard to then do all this complex signaling and so on that you saw in the drug effect modeling of the cell flow. So these are sort of complementary approaches depending on the detailed information that you would have. In this case, sort of us leveraging RNA velocity in this developmental process. So maybe I just do 5 more minutes wrap up, Pedro, and then I close. Is that okay?
Fantastic. And we can also open the floor for anyone who has questions. So please type in the questions or raise your hand and we can unmute you if you want to ask the question personally instead of writing it down. But
yeah, I think there's one question here in the chat box already. So from Wen-San Zhu says saying, to which extent can models trained on single cell data from established cell lines be expected to generalize to primary cells?
Oh yeah, absolutely. This is something that, a lot of pharma, I guess, bets on in the early screens, right? You always start the screen in cell line, and then, this is why I showed the organoids a bit. Then you go sort of a bit more realistic in vitro, or then you go to mice and you go to human. Like, how close is that stuff? I mean, what we saw in this thing, I don't even talk about cell lines, I just talk about these organoids now, right? So in this organoid atlas, remember, I showed this thing. So when we sort of really quantify how far that in vitro thing is to the in vivo situation, we sort of saw sometimes good, sometimes not. But because we quantify it, we can sort of optimize for this thing now. This is sort of what we do with the model, right? But yeah, cell lines are not perfect, but in cell lines you can do much more. So I think it's good if we can somehow integrate them. So if I may, I just want to have 2 points on perspective. So remember I talked about this perspective, about this virtual cell that a lot of people now think about. Can we do an AGI type of thing, like a language model on these cellular profiles? So there's been some excitement, and this foundation model, take all this data, train it once with a very big transformer, and then fine tune for downstream tasks, is something that there was a lot of excitement about. We recently reviewed this. I think the seminal paper is here from Christina Theodoris 2 years ago. A lot of people then following this up. Bo, for example, with his SCGPT and so on. These things often work quite well, but then also sometimes are really not yet outperforming more specialized models. So I think it's interesting where this goes. We've been doing our own, which we call niche former, which takes disassociated as well as spatial data, is fine-tuned for sort of niche prediction. I won't show you the results now because I don't want to go over time. I just want to sort of show you one point that we sort of compared to many sort of classic linear and other approaches. And we did very detailed benchmarking, and we see that here lower is better. This model is actually doing a good job at a hard task. So I think the problem we have in the field is that like cell type prediction is not a hard task. We need to come up with harder tasks for these models. Like, we don't have the benchmarks yet that, language model, natural language models have with, passing the bar or whatever, right? So I think it's interesting times where this goes. This is coming out soon. My very last point, let's say we have this model. This could be this multimodal foundation model. It's a big transformer. It could be sort of these more specialized diffusion models that I showed you before. I think what's exciting about it is that we want to put this lab in the loop, right? You want to do experimental design. So we propose an experiment, we go back. I think this is where things go. We wrote this a bit in a perspective, check it out together with Bo. But I think sort of coming up with the math to do this next proposal. Like, do we want to minimize uncertainty? Do we going to go for a specific target? Do we come up with more fancy stuff? Do we do batch design? I think there's exciting questions here and my lab, and so we're working on that. So to summarize, I showed a bit about how to learn atlases. I presented Cellflow and Velo, as well as shortly niche former. So the database, the hybrid model, and then the transformer-based ones. There's sort of various trade-offs where we go. I think scaling is going to be exciting, and we want to be able to act upon. I think I acknowledged all my people already, but this is my lab, Bavarian Mountains. If you want to come by, say hi, and really appreciate your invitation, and happy to take some questions.
Yeah, thank you so much. Any questions? You can just raise your hand or
yes.
type in. We have one.
Let me unmute you. There you go.
Hi, Fabian. Thank you so much for your talk. have a bit of possibly a bit of a curveball. So with your work, you've shown that it works both on single cells and their sulfates, and also on organoids. I wondered if you think in the future it would also possibly work on more sort of meta-genomic, meta-transcriptomic data sets as well. I ask this because my background and what I'm working at the moment is sort of the gut microbiome, and it's really, really difficult to try and predict how treatments for people with different digestive diseases, how their bodies are going to respond to, say, probiotics, prebiotics, to fecal matter transport, like a whole bunch of different things. But because it's such a big variable system, trying to predict what interventions will affect people is still a massive problem in this field.
Yeah, I have 2 points. Thanks for raising this. I mean, naturally, right? And that's not something very single cell specific to that. And we've been trying to go a bit beyond just the cellular thing. The last example I showed in zebrafish, that wasn't an organism, right? So, there we could show that, and quite honestly, albeit on a very sort of big effect type of situation, I mean, namely organism development. That's sort of where you really see big effects. That this model was doing a quite good job. We actually saw also with cell flow that the more subtle the effects go, the harder these predictions were. And there's one big sort of set of perturbation effects that even without microbiome we have, like environment is going to be a whole new thing, but even within humans, namely genetic variation, you would want to put into your model, but these effects are often very subtle.
Yeah.
Like, it would be cool. We don't have yet enough data, but there's new stuff coming out. I mean, UK Biobank, one of the best resources for human variation. Nicole Soranja has been starting a project called Cardinal with more than, I think, 5,000 people now doing PBMC things on a seq, and we're involved in the analysis there. And there you can sort of start asking some of these questions, but yeah, I think it's a bit early times. And then now you adding environment, I think it's getting even harder. Let me just put this one paper in that I'm sort of a bit bullish about. So we have this pure black box prediction thing where we just sort of add a whole bunch of existing data sets together. Again, mostly saline, but more recently also organism. I just put it in here. And there, this is not specific to single cells. So it's like a general perturbation effect thing. We sort of generalize any type of perturbation experiment as sort of a little sentence, GPT-like if you want, and then we sort of able to predict any other new sentence across different phenotypes. And for example, there we were able to look. I think we don't have in the paper; it's in the revision at the moment. We weren't allowed to upload it. But there, I think we looked at melanoma cell lines, something like that, and went to all of Campbell and selected then out of this sort of prediction in the design fashion 5 things that we want to follow up, 5 drugs that we want to follow up experimentally, and 4 out of those 5 actually had an effect. This was like huge. Typically screens give you very little effects, and I think that could be maybe tried out in your situation as well if you could potentially fine tune it for sufficient data. So maybe check it out, or maybe get back if you don't mind.
Amazing, thank you. I'll definitely look into it.
Thanks.
I do have a small question. So you did say most of these needs a lot of data sets, especially for the transformer based. Do you foresee trying to apply anything to reduce the number, having any any methods to reduce? Because for example, for gut microbia, sometimes you don't have a lot of, you just have patient data, and usually this is scattered and not very not, you don't have a lot. But I imagine using this model that you were talking to again for that.
Good question. I think quite honestly, we could hope that particularly in these single cell specific models, that including bulk information would be able to do a bit more there. And bulk, then you have in patients in much higher throughput, right? And maybe you can deconvolute or something like that, or you can sort of jointly learn embedding of single cell and bulk. There's some first people trying this. We have a few examples on that, but it sort of works to some extent. The other one is I think that's why I sort of also showed the hybrid model. I think this concept of adding everything in a very sort of data-centric model is cool. But benchmarks at the moment still show often linear models are still quite competitive, So like you have this super big transformer and then you don't need all the capacity of it. So I think having a simpler model often does a good job. And for us, then adding this very hard constraints, such as, adding, like having this prior from a GRN, which we know is not perfect, right? But it gives us A, more interpretation, and B, gives us more extrapolation power, at least at the moment. And I think that could be maybe something also in your case. Like if you have, let's say, some type of, I don't know, dependency of the embedding with respect to patients, you can sort of add this embedding model in here, and then you can learn from there.
Thank you very much. Any other questions in the room? So Jingong asks, for single cell perturbation prediction, several benchmarks have shown foundation model doesn't bring improvement over traditional simple methods. What do you think? Do you think model and data scaling will help?
Yeah, Jing, thank you so much for mentioning this. Was essentially what I was also just saying. So I agree. And that's why also the foundation model term. And in this case, I always call them transformer-based models, right, Jing? Because I think you're mostly talking about this. Yes, those embeddings actually didn't often help. And it's kind of sobering, right? That's why, I think the tasks that are being asked, you need to check it out. Those tasks are often very simple. It's just like cell type prediction. And, we had a paper, I think 5 years ago, that was saying, hey, deep learning is not needed for cell type prediction in an organ. A linear regression, like a logistic regression, literally is enough. And nowadays also there, this is sort of just a repeat of some of these things. But with more data size, nonlinear effects will be much stronger. So I think it's just a scaling thing that we haven't seen yet. And B, I think also the way how we build these models maybe is not the most clever thing. So those, for example, obviously we don't have sequential data, right? And if we talk about gene expression or protein expression, so there's no sequence. I mean, these things are sort of a big graph or something like that. So just treating them as if it was sequence, and there's tricks such as ranking and so on that sort of adapts the transformer formulation is maybe not the most clever thing. So for us, this flow-based formulation, that was really helping out. And there we show that we outperform linear or additive models.
We do have another question from Dr. Fabian Theis, someone who was logged in as you, but is AI VC can help for some Mendelian disease model and provide new therapy? For example, some cystic fibrosis patients with complex variants of CFTR doesn't respond to CFTR modulations. Is AIVC can help propose new therapy?
That's a cool one. I really think we need to add genetics, like, and you have even Mendelian disease model, like like a single knockout or so. So for that, yeah, this has to be possible. And I think maybe it's really the right way to start. Like not complex disease, like all little things, but let's say there's this really very close, clear thing. It's a Mendelian type of effect. Can you put this in the model? I haven't done that yet. I think because we have these embeddings, I think it's maybe not so hard to add genetics just to whatever thing we do, maybe just as a covariate first. But in this case, it should be able to do something like that. So if you or if anyone is aware of sufficiently large, particular single cell observations, which we typically use for this thing, I'd be very happy to take a look. And I think this will happen. Yeah. So very, very, very interesting point. I think really people go into this.
There's a follow-up question from Jiayang Zhang. For the underperformance of foundation models, did you mean that the model architecture is not good enough? Like, should the hybrid like what you mentioned earlier with the understanding of biology mechanism?
Yeah, I think at the moment, actually, it's kind of true. So, if you want to show that foundation models are good, like even by not comparing to other, and it's often, if it's a prediction problem, it's hard to beat XGBoost, right? And we need to benchmark across that type of stuff. It's not as everything is deep learning now, right? So, I think what you want to do is what I think people did in OpenAI in one of the seminal papers with the scaling laws. Like you want to show that if you sort of increase data from 1 to 10 to 50 to 100%, you see sort of a systematic increase of the model eating up the data. And we had one of those first papers trying to do this actually for this sort of somewhat simpler task of cell type prediction. We call it SE-TAB. I just put in the chat here. And maybe I can just share the screen if you don't mind. I don't want to take too long. Really, just briefly. But you asked about the scaling law, and there's some new papers now coming up with scaling laws. But one thing we did in that paper is here. So this is the model. It's just like a bit more fancy, attention-based supervised model. But I just want to show you this plot here. So this is plot 2, and this is where we show not trivial scaling behavior. I mentioned that cell type prediction is boring, but we have cross-tissue cell type prediction, and then we see that if we do subsampling from donors, we this thing here. So if we now increase the number of cells in the training set, any linear model, and this was at a very simple baseline, it's just necessarily thresholds out. So it doesn't have more capacity. So it eats up every capacity it has, and then sort of that's as good as it gets. And the other thing sort of keeps scaling. So here you would be able to say, hey, yeah, we need more donors, we're gonna get better. And I think this is what we're missing a bit yet in our field for more complex things, like for foundation. In this case, it was just for cell type, but like a foundation model, and you want to see that it sort of gets better and better at the downstream tasks, the more data it eats. And I think with these very big perturbation models, this is starting to happen. So, I showed you very briefly for cell flow that it starts doing that, but cell flow is not yet a foundation model, it's just straight on one data set. So we're currently doing that. Cool.
Well, let's do one last question and then we can let you go.
No worries.
Which type of data gain do you think it is better for modeling perturbations? More post-perturbation profiles per perturbing condition, or more perturbation
I
conditions?
like that a lot. Really good stuff. Like this is, you guys are in the design topic, right?
Mm-hmm.
So, how do we design an experiment? Like pretty much most of my experimental collaborations, I think including the UK Biobank, typically we design an experiment this way that we say, hey, we have X money, let's just do as many samples as we can get for that money, right? But even then, there's a trade-off. Like, should we do a deeper sequencing of one cell or should we add more cells? That's a very classical one. And we actually, there was like a junior PI at our place, he had a paper called SE Power, which is just like a power analysis for this differential expression question. And an interesting finding there was, it is better to do more cells than deeper sequenced less cells. Okay, so that's this trade-off. And now you're asking, unfortunately, can't pronounce your name because I can't read the Chinese letters. But what you asked there is, should I go for more profiles, maybe more multimodal, maybe sort of more time points or something, or more perturbation conditions? And one thing we saw on the SETAP paper, at least, was it's better to go for more perturbation conditions. Okay, in this case, it was just donors. So that's why, the Tahoe data set that you might have seen, that sort of has quite a few. I think it's like 100 million cells, and this sort of has really across, I think, 1,000 conditions or so. This is getting, I think, really exciting. And I think ideally increasing that would give us more than cells, but we don't have an experimental design written up for that. We're trying this a bit. I'm sure there's others on it, but you want to be quantitative about it. But it's very hard to come up with a statistical model for that. So. Okay, thank you so much.
Yeah, thank you. Thank you for the interesting talk and like answering all the questions. I think the audience are really happy. Yeah.
Yeah.
Very interesting, Doug. Thank you for your time as well. And thank
really
you
nice
everybody
to see how
for
you guys organize, and yeah, big hi to our friends in the UK. We're sort of moving a bit closer, right? We need to make sure that we keep the science connected, and happy to do some exchanges or follow up on this. Take care.
Yeah, so please keep an eye on our website because this will be the final one for the whole year, and we're planning the new this seminar series for the new academic year very soon. So yeah, thank you and bye.
Bye.